Hello, my name's Aminat Lola Shalebo, but my patients call me either Dr. Lola or Dr. Lola, depending on which languages they speak. I'll answer to either. My patients are children with potentially blinding disorders who are looked after at Great Ormond Street Hospital, where I work as a consultant paediatric ophthalmologist. I'm also a researcher at the UCL Great Ormond Street Institute of Child Health, and I'm funded primarily by the National Institute for Health and Care Research, or NIHR, which is sort of like the research arm of the NHS. I'm also lucky enough to have been funded by other groups, which are shown here on the first slide, and include the Academy of Medical Sciences, Fight for Sight, and the Wellcome Trust. We need to get better at picking up and monitoring a potentially blinding childhood eye disease called uveitis. Uveitis affects thousands of children in the UK. We currently look for and monitor uveitis using a microscope called a slit lamp. The doctors use to look inside eyes and count the number of inflammatory white blood cells they can see floating around inside the eyes. These white blood cells are in spaces where they should not be, and getting rid of them involves using really quite powerful medication, which children usually inject themselves with every week to keep the uveitis away. There I am using one in the picture on the left-hand side, and in the pictures on the right-hand side, there are the magnified views that I have of the front chamber of the eye, which is, in a child with uveitis, full of inflammatory blood cells or white flecks floating about in what should be a quiet space. If you get two specialists to look at the same eye, they only have about a 60% chance of agreeing on how much inflammation they're seeing. Also, it's likely that knowing what types of cells that are floating about inside the eye will tell you more about that child, or even that adult's, because it can affect adults, uveitis. We could get a sample of this fluid in the eye using a needle, but that isn't the ideal step, as you can imagine. So we've been using OCTs, or optical coherence tomography cameras, to get a better look at eyes affected by uveitis. Here's a magnified view of the inside of the eye using an OCT scan. We're getting so much information, and we know that these scans are really sensitive and they're able to see more and measure more than a human looking inside the eye the routine way could see. We also know that, to some degree, the more images you can get from the same eye, the better an idea you have of what's going on inside the eye. But we also know just how long it takes for humans to look through all those images in order to analyse them. This is what we do for each image we get from the eye, and we can get hundreds from each eye of each child. An image grader assesses the quality of the image. Is it good enough to tell us what we need to know? Then they draw the region of interest, where in the scan we should be looking for cells in the image, in yellow in that diagram. The image is then changed from shades of grey to pure black and white. We call that thresholding, helps the cells to really stand out. We then use a computer program to detect how many cells are in the region of interest. It can take quite a while. We're building a system to do this automatically, but we need to teach that system using images that have already been labelled by a human. And that's where the Zooniverse Citizen Science project came in. We asked citizen scientists to look through images from children with and without uveitis and help us with different analysis tasks. One of these was judging the quality of these images as good, poor, or limited by some sort of artifact or image error. We also asked them to tell us what sort of image error they could see. They classified artifact or image error as being due to movement. So in this image, there's been a small movement during the scan, and that's why there's that step in the structures in the middle of the image. They were asked to tell us if they saw lots of movement, and also these white splodges, which you can see in this image, which are due to eyelashes. Sometimes sometimes the eyelash artifact is quite subtle. So for example here, where the eyelash splodges are quite small and appear to be coming from inside and not actually outside, which is where they really are, of the eye. Sometimes you can see this flare or glare signal, which is due to an aiming light from the OCT camera shining on the eye in just the right or wrong place. Citizen scientists were also asked to draw the region of interest and to tell us where they could see any cells in the image. The project went live in June 2022 and we had a great deal of interest, and we are so grateful to the almost 3,000 volunteers who helped us to label these images. So what are we finding? Well, this is a confusion matrix, a way to look at findings from what the most popular response from the citizen scientists was and compare it to what the experts said about the images. Okay, so of the 313 images that the expert said were good, so that's the first column with all the numbers in, most of the citizens, 272, the number in bold, agreed, although some said that the image was limited. But crucially, they didn't say that a good image was poor. And of the 278 images that experts said was poor, citizen scientists from Zooniverse agreed on 209 of those images and only completely disagreed, that is, said that a good image was actually poor 8 of those 278 times. We also looked at how much agreement there was on the type of artifact present. We used something called a kappa score to measure agreement. So a kappa score of about 0.61 to 0.81 is substantial agreement. Anything over that is almost perfect agreement. Citizen scientists achieved substantial agreement with experts on almost all of the artifact measures. The next step is looking into possible reasons why some citizen scientists perform better than others. Do they learn to do better over time, for example? Does the type of image they're seeing help? We're also completing some analyses on the agreement between citizen scientists and experts on that region of interest that they drew, and also the cells they're seeing. We're already pretty confident, though, on labels about quality and artifact. And once we understand exactly how reliable the other labels from citizen scientists are, we can really superpower our work on using this OCT imaging to improve outcomes in this blinding childhood disease. Thank you for your attention and your interest. And if you're one of our Eyes on Eye scientists, thank you for your time. I also need to acknowledge my funding sources and my wonderful collaborators pictured here. A longer, more detailed version of this video is also available, and you'll find it where you found this video. Thanks.